Hello, everyone. I am Kenny Coogan. I'm the Education Director for the International Carnivorous Plant Society. And thank you so much for attending this live webinar. Today, we are joined by Kadeem Gilbert. He's going to be talking about the evolution of Nepenthes pitcher plant diversity and their interactions with symbiotic insects and microbes, because Nepenthes do not eat everything. Sometimes they provide little homes for organisms, so that's what we're going to learn about. And after that, I'm going to do a little summary about the International Carnivorous Plant Society. And then after that, Kadeem is going to take your questions. So if you're attending this live, just go down to the chat and feel free to type in your questions, and then I will funnel them to Kadeem at the end.
Great. So thanks for having me. I'm Kadeem Gilbert. I recently started a position as an assistant professor at Michigan State University, based at the Kellogg Biological Station. And I work largely on Nepenthes, but I am overall interested in symbiotic interactions. And I'm going to run through basically all of the work that I've done with Nepenthes, especially, and thinking about these questions with symbioses. So as you can see here, pitchers they're a bit of a paradox in that they're not just this pit of death, right, where we see drowning flies in the bottom like this. But when you peer inside, you can also see things like these. So this video that's playing right now are some phorid fly larvae that I collected from a pitcher of Nepenthes gracilis in Singapore. So you can see them out there in a petri dish eating a mealworm. I'm really fascinated by symbiotic interactions. And then I'm also really overall fascinated by trait diversity. So the genus Nepenthes, as you may be aware, is this really diverse group of carnivorous pitcher plant with over 170 described species. I have to check it periodically because then there's so many new discoveries being published every day as people get out into more remote areas. But across the range of its distribution, you see a range of morphological diversity, right? So lots of different species with their pitchers of different shapes and sizes. And then in some cases, researchers have found connections between that morphological diversity and ecological implications for their interactions with other organisms. So for instance, we have Nepenthes alba marginata, which has this ring of white trichomes along the collar that it uses to attract almost exclusively termites, in contrast to most other Nepenthes, which largely eat ants. And then, oops, went to do that too fast there. You have Nepenthes ampullaria, which has evolved to be an omnivorous plant, right? So with its reduced lid, it is able to collect leaf litter from which it also gets nitrogen. And Nepenthes lowii, which has one of the most bizarre relationships in the entire plant kingdom, where it's literally evolved into a toilet. And this shape allows a tree shrew which is attracted by this nectar that's secreted on the lid, to sit astride on the pitcher, where the plant is able to get pre-digested nitrogen. So in addition to these, the vast diversity between species in Nepenthes, intraspecific or within species diversity is also quite ubiquitous. So as you probably aware, almost every species in Nepenthes has this dimorphism where they produce both lower and upper pitcher traps, which even on the same plant are distinct from one another. And between species, you can kind of, you have these similarities wherein the lower pitchers have the wings, but then the upper pitchers do not. And also the difference in the climbing tendrils, where you have kind of more gravitropic, so like a downward growing tendrils for the lower pitchers and these twining tendrils for upper pitchers. So there's this really interesting level of diversity in shape between and within species. And then I'm interested in questions in understanding why, how does that functional diversity contribute to interactions with other organisms? So in some work that I've done a lot of work in Singapore, where there's 3 species, as you can see here. Nepenthes gracilis is one that I've done a lot of work on in the natural parks in Singapore. And this is a great species for looking at questions about intra-specific diversity, because it exhibits all of that diversity that I've run through, where you have the pitcher dimorphism, but then also this really interesting color dimorphism that really stood out to me the first time in the field, where some pitchers are a lot more red and others are basically green. But even better than that is the fact that these different variants of pitchers can grow right next to each other in the field. So this lends itself to a nice, simple in principle, natural experiment. So one of the things I did is I went out to these plots in Singapore, and I had emptied out all of the pitchers in little patches, and then allowed them a month to start collecting insects anew and also becoming colonized by symbionts, so I could see in a more controlled way if differences in the characteristics of the pitcher lead to differences in their prey capture or in their symbiont colonization in the same areas. So here's just an example of what it might look like to empty out a pitcher Nepenthes gracilis. So this is an image I took, and you can see a lot of disembodied arthropod parts, largely ant heads, but then every now and then you get other prey items like the pseudoscorpion here. Wings are also slow to be digested, so able to get a count of those and get a rough count of flying insects. And here is that phorid, specifically Nepenthes is the genus, the same one from the video at the start. So right, I did that. I emptied out my pitchers, so I can directly compare the rate at which pitchers that vary in different properties, either their color, the pitcher morph. Another thing that I looked at was connectedness, which is a metric of for upper pitchers, how interconnected they are with surrounding vegetation. Perhaps species that are in pitchers that are more intertangled with vegetation might draw more ants as they use highways. So that was one idea. Also measured the pitcher size, and I looked at how far it was from the ground. Because then we have upper pitchers that are a little high up, but then some that are hanging kind of closer to lower pitchers. But after after a lot of effort, and then this was one of my first projects as a grad student, I spent so much time under the microscope counting all of these different arthropods. And what I found in the end was basically none of the traits that I had measured really had any significant difference for any of these insects. So that basically the only thing I could say for sure was that larger pitchers caught more ants, which was not entirely exciting. But while this was kind of discouraging, I was drawn to continue probing this question. So why is there so much of this diversity if it's not related to how they're interacting with the insects? And in particular, I was interested in this color difference because we believe these pigments, anthocyanins, come with a cost. They're they take energy to produce. So why go through the energy of coloring your pictures if it's not really helping with prey attraction or signaling to the insects in some way? So yeah, right. That was the first question. We know that anthocyanins signal to, say, pollinators for red flowers and that sort of thing. But I didn't really see that here. However, there's lots of other hypotheses for the functions of anthocyanins in other plants that we can explore as well. So one popular idea is that there's physiological functions that are related to photosynthesis. So I will go right into that. So if you spend any time in the tropics, you've probably seen trees like this or other plants where the young leaves at the top that are directly exposed to sun are red. And then when they become mature and fully expand, then they turn green. So there's some evidence that anthocyanins can act basically like a sunscreen to protect the leaf from the harmful effects of UV radiation, either by directly shielding their photosynthetic pigments or by reducing the negative effects of the byproducts from too much UV stimulation. But basically, this has been seen in other plants. So my next question was, is this the case in Nepenthes? Maybe it's really not a signal for insects, but rather has this kind of physiological function. So to do that, I went back to my same plots in Singapore, and I estimated how much canopy coverage there was over each of the individual plots. So yeah, basically just took an image like this and then calculated the white area. And I did find quite a strong relationship, as you can see here, between the proportion of red pictures at a site and how open the canopy is. However, the trend is the complete opposite that we would expect from this photo protection hypothesis. As you can see in these darker areas, so more canopy coverage, greater proportion of red, and as it's more open, greater proportion of green. So a strong trend, but complete opposite from what I was expecting here. Not only that, but remember how there's upper and lower picture morphs. Well, it turns out that the color dimorphism is disproportionate. So upper pictures, which are by definition closer to the sun, especially once they are climbing up into the canopy, those are almost always green. Very few of these upper pictures of gracilis are red. Furthermore, some just observations in the field, it's right, if you find a red picture, it's going to be a lower one. And then a lot of these are often completely varied. You find pictures that are underneath the leaf litter like this or like this, and they are fully red, without being exposed to the sun whatsoever. And then you can see here, this is has not been opened yet, so they're forming red as opposed to turning red. Oh yeah, and then here's your example of the upper pictures in the trees. You could see it's sunny, and it's almost kind of hard to pick out the pictures, from the surrounding foliage. Right, so I kind of discounted that first hypothesis of visual signaling, and I also didn't see in effect this physiological function that we expect from other plants. However, there is a third idea about anthocyanins that's in the literature as well, which is not as popular, but there is some evidence for this as well, that it could be, rather than signaling to pollinators, say for instance in flowers, that red leaves may have a defensive function. This was especially well shown with autumn coloration in oaks wherein the red color acts as a deterrent to aphids, basically telling them this leaf is no good, it's not tasty, you may as well not try. So could that be the case in Nepenthes? So like you, this might be the last thing you think about with carnivorous plants, is thinking about herbivory. It was certainly the last thing that I thought about, but just like any other plant, it's not as common, but you spend enough time in the field, you see that they do fall prey to herbivores as well. So again, going back to those patches in Singapore, I decided to quantify this, and I found indeed, it turns out that the red pitchers in the same patch are attacked disproportionately less than the green pitchers growing around them. While not necessarily conclusive, this was some evidence really pointing to the possibility of a defensive function in Nepenthes, which kind of goes to show how traits are multifaceted. While a pitcher may be for the purpose of carnivory, they are also a leaf, and other things may come into play. But that said, I'll continue along this line a little bit more, as I've just shown some data from the field and with Nepenthes gracilis. This is just one species, so I was curious about how these traits, in particular this coloration, how it relates to the genus as a whole. So to do that, I needed to take a phylogenetic approach. So I used the then publicly available sequences of Nepenthes species that were available on GenBank, the public genetic database, and reconstructed the phylogeny and scored traits using McPherson's pitcher plants of the old world. And I scored a bunch of different things, including qualitative traits like different coloration features, and then also morphological traits like length of the pitcher, length of lamina, and so forth. And I won't go into all of this today, but I'll just hone in onto this story of color polymorphism. So I'm defining color polymorphism here as the differential pigmentation between upper and lower pitchers on an individual plant. So what I did in a phylogenetically controlled way is I did what's called stochastic character mapping to understand the probability of switching between different states. So we have 3 states here. Either your polymorphism has the lower pitchers as more red, or the upper pitchers as more red. And I mean relative to the level of pigmentation to the other morph. Or you lose your color polymorphism. And what I found is that there's a heavy bias across the genus towards the redder lower pitcher state. And then you are far more likely to lose your color polymorphism than you are to evolve to the other direction where you get red upper pitchers. And I'll wrap this up with some more findings on the evolution of color polymorphism, which I believe links back to this possible relationship to herbivory. So in addition to looking at the color polymorphism, I looked at their growth habit. So your typical climbing species like this, you see as I just presented, where the arboreal pitchers begin to go more green compared to your red lower pitchers. Very interestingly, I found that the evolution of an epiphytic habit was correlated with a loss of color polymorphism. And then the reason why I liken this to more indirect evidence as to a relationship with herbivory is that in studies of other climbing plants, we found that climbing acts as herbivore defense in a way. So if there's a gradient of herbivory pressure with greater herbivory pressure on the ground that a climbing plant can escape by growing up. So if you force a vine to grow prone, then herbivory will increase. So if this is the case, then what we're seeing with coloration makes sense. If this is the case, and if that red pigmentation is in some way related to defense, then there's greater selective pressure for lower pitchers to become pigmented, right? Whereas there's differential selective pressure on the plant in your typical climbing species. But because an epiphyte starts arboreally, both pitcher morphs are in the same microenvironment, and thus there's not this differential herbivore pressure. So if that makes sense, that is what I'm thinking is for the evolution of color polymorphism in Nepenthes here. So I'll just wrap up this section, but before doing that, mention that now, so I've started my lab last year, if I didn't mention that. I have one grad student now who's interested in questions about anthocyanins and Sarracenia. So we'll stay tuned and we'll learn more about this group as well. So for the rest of the talk, I'd like to focus on that main interests I have on the living components of the pitcher community. So I have up here a definition for symbiosis, because different researchers will use it in slightly different ways. For me, symbiosis is not a synonym of mutualism. It's relates more to the physical association between the plant and the organisms living in or on its leaf. So these can either be parasitic or mutualistic. So parasitic in the case of things like leaf miners or galls, and mutualistic like your ant-defended acacias or Myrmecodia, which produces these ant houses inside for getting nitrogen from ants. But the interesting thing that links both ends of the parasitism to mutualism spectrum conceptually for me is the fact that in both cases, we have to think about the physiology of the plant, because these insects or other organisms are in the environment of the plant has the ability to have an outsized effect on its inhabitants by changing its physiology and therefore changing the entire environment that its symbionts are exposed to. So that's a concept that I'm really interested in here. So in this first case, I'm going to tell you about work I did in the context of a common garden. So this came about from collaboration with growers in Singapore, and the Hort Park Nursery. They were growing many different species of Nepenthes that were being grown to be put into the cloud forest dome at the Gardens by the Bay. And then if you haven't been to Singapore, I highly recommend going to Gardens by the Bay. It's a great place for botanists. There's a lot of botanical attractions there. So while these plants were being grown for horticultural purposes, it turns out a lot of features of the setup were really great for a research question that I came in to propose that we look at. So these plants are in a common garden, as I said. They're all around the same age, grown clonally. The environmental conditions were very well regulated in their greenhouse, the temperature, humidity, and even more of note is that they had their own source of water, that they had a system in place for creating a set pH level, which they used for misting and watering the plants. And even more interestingly, they would also fill these pitchers with water, this common source. And remember that this water is starting around 6.5. And in 2 weeks, I sampled those communities, and then we'll look at how how microbial communities relate to the species identity in this greenhouse setting. But yeah, to go into that a bit further. So here's a sampling of some of those species that I sampled from. So you can see the morphological diversity. And like I'd mentioned, they started with water of pH 6.5 being put in. But then the really interesting thing is that there's physiological diversity between these different species. They alter the fluid properties in a lot of different ways. And most striking of all is how the pH changed in the time that the plants had to acclimate their fluid over 2 weeks. So from 6.5, we go to this whole range of pH values. So some species which are relatively moderate at pH 4 or 5, but then others which get down to 2 or even 1.5 in some cases. So some species are just able to acidify much more. So while the ability of the plants to regulate pH was not a new finding, Jonathan Moran's group had found that about 10 years prior to my study, but it's quite surprising to see how rapidly the change takes place and across a wider range of species here. So the question then, or my question is, so they differentially regulate their fluid properties, but does this lead to different microbial communities, even though, we're in this artificial greenhouse and they're just exposed to the same microbes? Do these different species cultivate a different microbiome? So to do this, we did this meta-barcoding approach. So basically focusing on a barcode gene, so one gene that we can use to identify microbes from all the DNA in the pitcher sample. So we take the fluid like this, and then target the 16S genes for bacteria and the 18S genes for the eukaryotes, and then assemble them into operational taxonomic units, or for our purposes, different species that we'll use to analyze the communities in each of these pitchers. So here's the result. So I'll step you through this, but in short, yes, the host species does play a big role. So what we're looking at here is a type of ordination. So we use these plots in microbial studies. Each point represents a community or a single pitcher, so the entire fluid contents there, and the proximity of two points represents how similar those communities are. So the closer points are together, the more similar they are, and farther apart, vice versa. And this is the same projection, just color for on the left and on the right. On the left, it's colored by species, and on the right, it's colored by pH. In this view, so the important things here are the stats, that both here are significant, that you can see in space, the different host species are separated. But then the key thing here is that if you zoom in closer and then you look, you compare across these, this green species at the bottom here, it occupies one particular end of pH space, right? So you can see how there's this gradient of pH where the less acidic it is, the communities converge to this point. What I'm showing here is that you have species differences in microbial assembly because different species have their characteristic pH levels. So pH, we know it's very important in any environment that we look in. Oops, that advanced on its own. Well, so yeah, whether you're looking at soil or in water, microbes, especially bacteria, are really sensitive to the pH levels of their environment. So so I'm going to go further into the importance of pH here, but I'll just transition by just briefly mentioning this latest paper that came out. But just to mention that there's variation within species as well. So going back to those species from Singapore, if you look over the life cycle of a pitcher, both Gracilis and Rafflesiana have this pattern where they start with a high pH before they open, and then they dip down into their characteristically acidic pH before drifting up by the time that they senesce and die. But Nepenthes ampullaria, interestingly, maintains its relatively high pH level throughout its entire life cycle. So I'm next going to talk specifically about the power of pH, but contrasted against some other external environmental gradients. So elevation, just like the latitudinal gradient, so you're probably aware of how at a large scale, biodiversity increases the closer you get to the equator. So there's a similar kind of universal ecological pattern with mountains, because elevational gradients, especially when you go from the tropics, you have these large climatic changes as you go up in elevation. And this has been a popular area of study, just as the latitudinal gradient has been for ecologists for centuries, basically, where we're seeing over and over again, whether you're looking at invertebrates or vertebrates or plants, basically diversity is going down as you go up in elevation. There may or may not be a mid-elevational hump, but we're talking about this general trend of decreasing. So this is just a meta-analysis of several studies, and this holds still today. On the other hand, microbes have really not been studied in this context for very long. It's only in the past 10 years that we've really had the technology to look at microbes carefully. So a lot of these large-scale ecological questions have not incorporated microbes. And then even more odd and frustrating for our understanding of microbial macroecology is that the few papers that have been published for microbes have found conflicting results. In some studies, they mirror what we see in macroorganisms, but then in others, you see an opposite pattern. In others, you see no change in elevation. And overall, in microbes, there's a question of whether or not they have spatial, if they have spatial structure in the way that multi-celled organisms do. So I contend here in this study that pitchers are a very useful system for looking at this question of how microbes react to these, let's say specifically these elevational changes. Because in the pitcher, we have symbionts in the same microenvironment, both the insects and the bacteria living together. So I'm interested to directly contrast whether these two very different types of taxa, are they responding to this elevational gradient in the same way? So for this study, I went to the Philippines. I did this work in the southernmost island of Mindanao, in the southwest peninsula here, specifically on Mount Hamigitan. So it's this 4,000 meter long transect going from 400 meters above sea level up to 1,200 meters above sea level. And I focused on collecting the pitcher contents from one species that was found along the entire range, so Nepenthes mindanaoensis. And I'll just show some pictures from the field so you get a sense of what it looked like here. So here's somewhere kind of low elevation, still relatively low, and then somewhere in the middle, and then getting higher up there. So the high elevation vegetation was characterized by these bonsai trees. So this is a fully grown tree, which never gets any thicker than about that because of the, the change in temperature and humidity and the wind kind of changes how plants grow. And here I am with R.C. Naive, who's a botanist from the area. So we went out in the field and we directly poured those pitcher fluid contents into sterile tubes. And then we measured some environmental, collected other environmental data as well, such as measuring the pitchers, noting color and noting the dimorphism. This is just for fun. See here, I found some cannibalism happening in the field that surprised me when I saw it. Yeah, so here's all that we looked at. So I have this broken up into external factors, which is that elevational gradient, which I should say explicitly, elevation is a proxy for climate, including temperature and humidity. And also measured that canopy openness again, and then we looked at some plant-regulated factors, so like pH, volume of the fluid, and the dimensions of the pitchers as well as the volume. So to set up these results here, here's some paired hypotheses. So first question is whether or not these pitchers are structured by elevation at all. So to my knowledge, no one had looked at this before, but so it was not a bygone conclusion that elevation had an effect on the biodiversity inside the pitchers, because maybe, as you can see in the alternate hypothesis, plant-regulated factors are more important. It only matters that you're in a pitcher rather than you're in a low or high elevation. Maybe you don't even care about the colder temperatures and high elevation. The other set of questions is whether or not microbes and multicellular organisms, or I'll just call them macrobes for short, whether these communities in the same microenvironments, whether they have the same response to elevation. On the one hand, we might think that they would, given that they're in the same microenvironment, but on the other hand, maybe some physiological difference can lead to different responses. So to go through these results, I'll say first that a physical count of specimens confirmed what we expect with arthropods, a lower count of all of these different types of arthropods as you go up with elevation, as we see time and time again in other studies. And for the bacteria and the microbes, including eukaryotic microbes, I did metabarcoding again, so looking at the DNA. I will also mention that this OTU approach was good for looking at the arthropods too, and also other animals. Like for instance, in this picture, I found frog eggs in the field, and that was also represented in the sequencing data. But rather than show you those ordination plots like I did before, I'm going to run through just results of this test of significance for these different variables that we measured here. So what we found is that out of all of these factors, for bacteria, pH is really only is really the only thing that's significant in structuring them. So elevation has some effect, but it's nowhere near as strong as pH for bacteria. On the other hand, both elevation and pH are important for the eukaryotes. So eukaryotes, like I was starting to mention on the previous slide, that involves not just unicellular things like algae and fungi, but also this arthropod and other animal DNA data is mixed up in there too. So one important question was, okay, maybe because we have these multicellular insects in there, that their DNA is kind of swamping the data. So I reanalyzed the eukaryotes without the multicellular organisms to understand really what's happening with the microbes. And it turns out, quite surprising to me, is that Unlike bacteria, pH was not really important to these unicellular eukaryotes, but rather elevation strongly was, just like it is for the insects. And I'll just run through some results for specific individual taxa, so specific species, right? So for bacteria, we see species like this, which you can tell from the names, Acidisoma and Acidicella. These are known to be acid-loving bacteria. And naturally, we see their abundance of their DNA decreasing as the pH increases. And that kind of a trend reflects a trend for this family. But then we have some other groups that are low in acid pitchers, but then high in more moderate pitchers. And then for elevation, some of the key taxa here are these algae. So here's an algal species that was only found at high elevation, basically, not found at low. And then the broader group to which it belongs, same pattern here. But then it traded off with this other group of algae, which were found predominantly low elevation, but not so much at high. And then I point out these algae because one surprising thing from the study is that going back to the literature, this exact trade-off in algae, these particular algal groups, you see in lakes. So in studies of high alpine lakes, this group is very common, whereas in low elevation or most lake studies around the world, you get a lot of these and then not so much of this. So it was quite surprising to see kind of the same big trends we see in actual full lakes, we're seeing reflected inside of these pitcher microenvironments. So to wrap this story up, going back to our hypotheses, indeed we found that communities in pitchers are structured by elevation, if we're talking about the eukaryotic organisms, right? On the other hand, for bacteria, plant-regulated factors appear to be more impactful. So this other set of hypotheses, yes, we're seeing different responses between microbes and macrobes, but it's even more nuanced than I anticipated, where we're seeing really just a difference between bacteria and everything else. And very surprisingly, algae and fungi operate more like insects in response to large-scale elevational gradients in comparison to the bacteria. So to wrap up this talk, I'm going to say that, so here we've shown both in the greenhouse and in nature how fluid pH is this really strong factor that influences these symbiotic interactions in particular. And then this led to a current direction that I'm currently still developing in my lab, an area began as a postdoc shortly before the pandemic and I'm continuing now. But how could it apply to other plants? So this question of pH, I will say that when you talk about roots or the rhizosphere, it's very well understood that roots alter the local pH levels of where they grow. And pH is also very well documented for below ground studies because it's important to structuring microbes in the soil. So that's very well understood. But on the other hand, I'm looking at these pictures, which are modified leaves, right? But turns out, oops, it bans on its own. It turns out, yeah, people have really ignored the role of pH above ground in the what you call the phylosphere on the leaf surfaces. Could it be possible that leaves, just like pitchers, can interact with external moisture, alter their external pH levels in some way that influences their symbionts living around them? And so, in fact, while it has been pretty much ignored, right? So in studies of leaf-associated microbes, you don't even see mention of pH in the literature. But there was a brief period of time, late 70s to the 80s, pretty much all agricultural studies where we've had some measures of pH levels on leaves. And it turns out that, yes, as expected, there's some evidence that it's not just these highly modified carnivorous plants, but that most leaves are able to some extent alter external pH levels. So for most plants investigated, you see this very, very moderate acidification in a matter of a few minutes, bringing the pH level down like half a point, and then it kind of drifts back up to neutral. But then, very interestingly, cotton, and there was also found other members of the family Malvaceae too, show this very interesting response where in that same time frame they rapidly increase the pH. So from 10 to 11 or so, which is really alkaline. So if you remember that pH slide from before, this is approaching bleach. So very curious, and no one had really attempted to answer or really had the ability to answer why, how is the plant doing it? Why is the plant doing it? How is it impacting its relationships with symbionts? So I'm looking across a diverse assemblage of plants here. So I have both ends of this pH spectrum. So the high acidity of the Nepenthes and the high alkalinity of Gossypium, and beet, which represents kind of your more normal plants. So doing two things. I'm looking on the plant side, how are they regulating their pH levels? And then also, how is that impacting interactions with microbes? So as we saw with the pictures, it's reasonable to suspect that different pH levels can lead to different microbial communities. I'll just briefly show this, that so before we knew something about the pH level of the digestive fluid, but to my knowledge, before I started looking at leaf surface pH, we didn't really know what's going on with the lamina. But it turns out that the lamina on Nepenthes can also rapidly acidify surfaces. So you can see here in this bicalcarata that it brought the pH down to around 4. So we're looking at the flat leaves and the lamina surface of Nepenthes. We have some physiological questions. So perhaps there's differences in the cation exchange in plants that acidify versus those that alkalinize. And in my current lab now, I have postdoc working in plant transcriptomes. So looking at all of the expressed RNA to look at differential gene expression, to understand mechanistically how plants with different physiologies, how they're regulating pH in response to external treatments, and also looking at how the microbiome approach changes with that similar approach. So that's what's going on now, and then I'll just close in thanking all the people who helped me along the way, and I'm happy to field any questions. So thanks for.
The 13th ICPS International Conference is happening in 2023 in Japan. We are excited because we've been to Japan before for our international conferences. The conference is hosted by many associations and societies, which are listed on the screen right now. So we appreciate all of these organizations for collaborating with ICPS. To learn more, you can go to carnivorousplants.org/about/conferences, or you can go to this link, which is what the Japanese Carnivorous Plant Society has created. But if it's too much, you can go to our website, and then we have the link there. We are going to be near this red dot. We're going to be near Himeji Castle. It's a couple of hours away from Tokyo, but Tokyo is probably going to be the place where you want to fly in. And then you have those bullet trains, and that's how you get to the conference. Pretty much everything is here, except for the very last day, which is a field trip. It's back over here. Here's an outline of the conference. For about 3 days, you have the conference, you have presenters, You have plant exhibitions and carnivorous plant sales from multiple botanical gardens and societies. Then on the 29th, we're taking a field trip to a botanical garden. On the 30th, we're taking a trip to another botanical facility, and we're going to visit some local carnivorous plants in the wild. And then on the last day, of course, this is like an add-on, but if you want, you can do a field trip to the habitat of a special pinguicula. Here are some photos of the beautiful facilities that we're going to be going to. This is where the conference is happening, and then nearby we'll be having a banquet. Here's one of the carnivorous plant, botanical, not carnivorous plant, but botanical gardens that has carnivorous plants. One of them just won the 2022 Guinness World Record for the longest pitcher, not seen here. And here's the facility. They have some beautiful rare Nepenthes. And we're going to be able to see a lot of cool carnivorous plants from around the world, but of course, we're excited for the ones that are native to Japan. All right, so these are the dates if you want to write them down. Basically, everything is due by March 31st. So if you want to register for the conference, if you want to go to the banquet, if you want to go to those extra field trips, everything is due March 31st. But what's really fun and interesting is if you want to be a presenter, you can present in English or in Japanese for 30 minutes. It has to be on PowerPoint. And if you present, you get to attend the conference for free. Something else about International Carnivorous Plant Society, if you go to the education tab, we have coloring sheets, activities, mazes, crossword puzzles. We have infographics, lots of great free educational resources for K through 12. Let's see. Something that we've been working hard on for 2022 is we created 5 different animated videos for beginners about how to care for carnivorous plants. You can check those out on our Facebook page or our YouTube page. And if you're at our Facebook page, or if you sign into the Club Express, you will see all of our upcoming webinars and happy hours, which we're always excited to host. All right, thank you everyone for that short commercial break. All right. So Jay has learned many things. Jay, we're happy that you learned some. Kadeem, Jacob was very excited. He told me a few days ago that he's from Kalamazoo, Michigan. He's so excited that you're in Michigan. He's at Western Michigan University. He loves plants. So Kadeem, I think your volume's off, but are you, you're not native to Michigan, are you?
Okay, there we go. Okay.
You probably get nervous. Okay.
Yeah, so I'm not native to Michigan. This is my first time living in the Midwest. I've been on the East Coast most of my life. But yeah, currently I'm living around Kalamazoo. So the field station is about 30 minutes from Kalamazoo.
How much research are you doing compared to teaching?
I'm doing mostly research. So this semester is my first time teaching here. I'll just be teaching half a course in the fall. So plenty of time for research.
Very good.
Oh, yeah.
I don't know if this really relates, but Jane wants to know, have you measured the photosynthetic abilities in red versus green pitcher plants? Or do you know, research from that?
Yeah, that's a great question. And it would definitely be important for understanding costs, like if the red pitchers are less photosynthetically efficient. I will say that I have not seen any data on that yet, nor have I attempted that yet. So it's quite complicated measuring photosynthesis on non-flat objects. I will say I'm looking into getting custom light core that would be able to look at photosynthesis in pitchers. I can't say that for Sarracenia that my grad student has begun looking at some measures, and as far as we can say right now, it appears that there's not a big difference in photosynthetic efficiency between red and green morphs, but that's very preliminary.
Benjamin wants to know, does lower pH pitchers digest prey faster or more completely or differently than higher pH levels?
Yeah, another great question. So the enzymes that they produce, particularly Nepenthes, which is to digest proteins, are their pH optima are at a lower pH level, so at a pH 2. So we do see, I believe other groups have looked at how rate of photosynthesis changes with pH level, but basically we could say that when you have a really mild pH level like we see in Ampullaria, that's associated with reduction in its photosynthetic, its digestive efficiency. I did see this also in the field, that was in that paper I just published. So that's why we kind of believe that that there's a trade-off, that the species which have a high pH, that they do so because they're outsourcing their digestion to their symbionts. So then that kind of balances out the cost of being less self-sufficient.
All right, I think we have one more question.
Put my email here.
Jacob is excited that you're so close and he wants to meet with you. And Kadeem, he's saying that his Instagram's name is Plant Prodigy, and everybody knows that. And if you're on TikTok, he's like reaching a million subscribers and fans on all of his channels. So he will be the future of plants and carnivorous. All right.
Yeah, we should definitely be in touch.
All right. This says, it's a bit unrelated to the presentation. I've had a Nepenthes for around 1 year and it seems healthy. Recently, some traps seem to be sweating on the outside, but it's cold here. Is this normal? Should I worry about it? Kadeem, do you have any insight into that?
My first thought is that is it sweat or is it nectar, like extrafloral nectar? Because a lot of species have nectaries not just on the lid, but then also outside of the pitchers. So if you just try to taste it, it might be sweet and it might be totally normal.
All right, well, thank you everyone so much for coming to this live webinar. We have, Kadeem, you can read in the comments later, but lots of people thanking you. Very nice talk, great talk, excellent talk. This will be posted in maybe a day or two on our Facebook and YouTube channel. And I encourage you guys to check out our future webinars and happy hours. We actually have one more talk in December. Then we'll just take a few weeks off and then we start again in January for the new year. And I really hope people consider going to the Japanese conference. We haven't had a conference in 3 or 4 years due to the pandemic. We're going to have a conference this May 2023. And then I think, this is a teaser for all you people who are here live, we might have a conference in 2024. And then we'll go back to the every other year. But I'm not going to tell you where 2024 is. So on that note, thank you, Kadeem, so much for doing your presentation. And everyone else, have a great day.
The International Carnivorous Plant Society wants you to be successful with your plants. We welcome growers just getting started all the way through professional scientists. We started an annual World Carnivorous Plant Day to celebrate these spectacular plants. Take a look around our website and you'll find historic documents about carnivorous plants, growing guides, free educational resources, and more. Have questions? Ask. We don't bite, but our plants do.